Welcome back to another episode of Science Outside the Lab, but this time I've taken the results from outside the lab inside the lab, and today I will be extracting and separating the pigments that I collected from the autumn leaves that you saw in the last video. So before I get into the process to separate the pigments, I will give you an overview of what happened in the last video. That is what happens to the leaves during autumn or fall. So during the growing season, leaves are packed with chlorophyll, that bright green pigment that plants uses to capture sunlight and to make food via photosynthesis. However, as the days get shorter and the temperature becomes cooler in late August into September, October, the leaves or the plants start preparing for winter. And how do they do this? They slow their metabolism and stop the production of chlorophyll. As such, the existing chlorophyll begins to break down and fade away. When that green pigment disappears, other colors that were always present in the leaf finally gets to shine. So the yellows and the golds from xanthophylls and the oranges from carotenoids, and sometimes reds and purples from anthocyanins are made anew. So the fall colors that we love are actually pigments that were hiding all along. Let's get into the experiment. In this video, you will see me use a simple lab technique called TLC, not the musical group, but thin layer chromatography. And I'll be using it to actually separate and see those pigments from the real autumn leaves. TLC works kind of like a pigment race. So if you think track and field, so each pigment or countries or track star will move differently depending on how much it likes the solvent, so the liquid, versus how much it sticks to the TLC plate. So think of it as a track. So it's how much it likes or it goes over the track surface versus how much like the spikes will stick to the track. So pigments that are more nonpolar, like carotene, will travel farther. So carotenes would represent Usain Bolt, big up Jamaica, while pigments that tend to be more polar, having more of a negative and positive charge at the end, like batteries, like chlorophyll B or the anthocyanins, they're slower, so they tend to be lower on the plate, like those other countries. I'll recap the technique, thin layer chromatography. As I said, it's a separation technique for mixtures. And so the pigment that I will extract from the leaves will move up a plate, a surface made of silica, with the solvent, the liquid, at different rates or different speeds to create a pattern of colored bands. So the key idea is that nonpolar pigments that do not have a negative or positive charge at their ends will travel farther. So they're the Usain Bolt, the Jamaica spring team. Whereas polar pigments travel slower, so they stay lower on the plate. You know, the coming last. So think of TLC like a race where each pigment runs at a different speed based on how much it likes the solvent versus the silica. Safety first. Because the solvents or the liquids that I'm using to do the separation, they are highly volatile and flammable. I am working in a fume hood. Should work in a well ventilated area. I'm wearing gloves because I'm working in the fume hood. I don't need to be wearing goggles because there's a glass shield. And I'm wearing a mask because I don't want to inhale the any escaping chemicals. And I'm using a beaker to act as my separation chamber. And I'm lining the glass beaker with filter paper, and I will pour just a small amount of liquid into my beaker, and I'm going to cover it with foil. The beakers don't have a cap, so I'm covering it with foil because I don't want it to escape. And what the filter paper will do will help to spread the liquid that will be my mobile phase into the entire beaker. So when I'm ready to put the plate in that has the pigment spotted on it, the extraction will be successful. So the solvents that I use, or you could use ethanol, you could use acetone, hexane, ethyl acetate, and some others, but it's always based on polarity. So you want to use a ratio or a mix of polar with non-polar to do the separation, because as I said earlier, the different pigments have different polarity. And polarity is just about the charges and your distribution at the end of compounds or molecules. So polar ones will look like a battery where it has a positive and a negative end, whereas non-polar one do not have any charges. During my walk, I picked up a lot of different color leaves. I couldn't help myself. They're gorgeous. And I tried to get different colors during different stages of the breakdown and from different tree species. So I have a green sweet gum leaf. I have a deep purple red sweet gum leaf as well. I have a maple. So if you think of the Canadian flag, a maple leaf, it's red. I have a yellow sweet gum leaf. I also have a orangish Japanese Zelkova leaf. To me, it looks like the Japanese cherry blossom leaf. I have a yellow cottonwood leaf. And I also have leaves from the oak tree. So let me walk you through what you will likely see happening in the video as I carry out this separation technique. So from the leaf samples that I collected, I'll take a small piece, cut them finely, and put them into test tubes. And once I've gotten them into test tubes, I will add the polar and non-polar mix of solvent into the test tube, and I let it sit for a bit to help with the extraction process. Once the liquid changes color, you could actually do this with ethanol, but ethanol is just not very good at the separation process, but it will extract the chlorophyll or whatever from the pigment, from the leaf. So once I start seeing that my liquid has started to change color, become colored, I will then add a pinch of sand into my mortar, add the leaves into the mortar, use my pestle to grind. So the sand will help with the grinding and extraction process. And I'll keep grinding. If I realize that my solvent has evaporated because they're volatile, I'll add more, and I might add more leaves as well and keep grinding, because what I want is a dark color. So if I'm extracting chlorophyll, which is green, I want it to be a dark, rich, concentrated green liquid coming out of the leaf. I want my yellows to be deep yellow. So those would be my xanthophylls. And I want the red or the purples that will be coming out of the leaf, if they are present. So the anthocyanins, I want them to be rich in color because that will make the separation better. The bands will be more visible on my silica plate. Once I've ground those and I've collected the liquid, the colored liquid, I let them sit for a bit because I don't want leaf pieces in it, and I also don't want sand grains. And then once I'm done, I'll go into actually spotting them on the TLC plate. So I need a pencil to run the TLC. Found one, don't have a sharpener. So the Jamaican way. Oh my God. Whoop. Ah, whoop. You remember, I used to do this in primary school. Ooh. Focus. Ta-da! Pencil is sharpened. Yeah. So I drew my line of origin using the ruler, and I've just slightly tapped with the pencil so I know where to put the samples. I'm going to put a code for different samples. I think I'm just going to go 1, 2, 3, 4. This is a test run, so hopefully it works. If it doesn't, I'll have to rework it, especially with the running solution. I'll take my samples and I'll use a pipette tip, so something with a small opening, or you could use a capillary tube to dip it into each of the different extracted pigments that I have, and I'll tap them along my line of origin, which is about 1 to 2 centimeters away from the bottom of the silica plates that I'll be using. And remember, I drew it in pencil because if I used a pen or marker, those instruments contained pigments and then those would get separated. And I don't want the marker or the pen to obscure my results. So that is why I drew my line of origin in pencil and tap where I'm going to put my pigments on the line of origin, and I'll tap. I'll let the silica plate dry in between. If it looks too pale, I'll go through and add more. I don't want the taps or the points to be too big, but I'll keep doing it until the colors are bright and vibrant. And once that's done, I'll put it into my extraction chamber. Anyway, so once the plate with the spots have dried, I will put them into the different chambers that contain different ratios of polar and nonpolar solvents or liquids. And I will then put the plates, I'll try to get them upright into my beaker or chamber and cover them and watch the liquid or the solvents pull and separate the different pigment spots. And what is expected would be different colored bands going up the paper or the plate as the solvent pulls them up. So the separation wasn't perfect. Nothing in science is perfect. So I'm going to draw what one would expect from a successful thin layer chromatography. So pretend this is my silica plate and I draw my line of origin where I'm going to put my spot. So I can do 1, 2, 3, 4, 5, 6, 7. Perhaps I should have had such a big space. And then at the top, I'll put my solvent front because I don't want the bands to run off the paper. As a recap, I did a colorful experiment to visualize why leaves on a biochemistry basis, leaves change color in autumn, and I separated the pigments using thin layer chromatography. So why do leaves change color? Well, when autumn arrives, or fall, you might call it fall, the days get shorter, the temperature drops, tree metabolism slows, and so the trees stop making chlorophyll, which is a green pigment. Chlorophyll breaks down and fades away, revealing other pigments. So you'll have your hidden yellows and oranges showing up, and sometimes red and purples are made fresh or new. So let us meet our pigments. From chlorophyll, you have chlorophyll A and B, and they tend to be green or blue-green, and your role is for photosynthesis, and chlorophyll is broken down first. Next, we'll have the orange or the carotenes. Those are always present in the leaf, and they're nonpolar. We have our xanthophylls, which are yellow, and what they do is they help the leaf absorb excess sunlight, a bit like a UV protection. Next up, we might have our red purple from anthocyanins, and these are only made in autumn, and they also help in protecting the leaf. So these main pigments should have been separated because each will have a different polarity, and so they'll travel at different distances up my silica plate. So let us talk about what's going on here. So, as I said, now we've completed the race. It's time to get our medals. And who's at the top of the podium? And we're giving out medals for first, second, and third: gold, silver, and bronze. So in first place, the Jamaica sprint team, like our Usain Bolt or Shellian or Oblique, etc., they would be getting gold. So they would be the carotenes in our example here. They travel farthest up the plate. They're non-polar, right? In second place, it might be the US team because that's up. It's always a battle between the US and Jamaican team. So they might be the yellow, the xanthophylls, the yellow gold. So they will get the silver medal because they're next up. So they're a bit more polar than our carotenes, which are non-polar. Next up, in our third place, would have chlorophyll A. So it's blue-green. If you think about cyanobacteria, also known as the blue-green algae, contains chlorophyll A. So they're blue-green. So they are much more polar than our carotenes and our xanthophylls, but more non-polar than chlorophyll B, which is our yellow-green. So first place, carotenes, orange, always present. Second place, xanthophylls, our yellow and our golds, which absorb excess sunlight. So they gets the silver. And then in third place, we have chlorophyll A, or blue-green. And I said it's in that blue-green algae, the cyanobacteria. And I mean, they don't medal after third place. But in fourth place, you'd have our chlorophyll B, which is yellow-green. And I want to point something out. So a leaf might appear yellow, right? But upon separation, it will reveal orange because the orange or the carotene is always present in the leaf. And the other colors or pigments that wouldn't medal, you would have your phyophytin. So they tend to appear brown, and it's because it's the graded products of chlorophyll, and they stay close to our line of origin. So they didn't leave the block, basically, or they barely left the block when the gun went off in the race, the starting block. And then the one that is really, really, really tripped up at the block would be our anthocyanins if they are present. So our reds and purples, they are very, very, very polar. So they don't move very far up our silica plate. So yes, these are our pigments. And I did this far. Thank you. And I have some questions for you to answer in the comments. Why do pigments travel at different distances? Which pigments were already in the leaf? Which pigments are made new or fresh in autumn? And do you have a favorite color fall leaf, and which is it? Thank you for stopping by, and I hope to see you in the next video. Ciao!